5, we're going to continue our unit on biotechnology. And we're going to start off talking about genetically modified organisms, or GMOs. These are organisms that have acquired one or more genes by artificial means rather than by traditional breeding methods. And again, when we're doing things with genetically modified things, we're going to use plasmids, or those circular piece of DNA. Okay, there's a lot of genetically modified organisms, bacteria, plants, animals. Okay, so the transgenic organism is an organism that has acquired the gene from a different species. Okay, and this is just how this process is occurring. Again, it's dealing with the plasmid. So the desired trait or gene is inserted into the plasmid. It's a recombinant plasmid, again, combination of two different DNAs. And then you put it into the plant of choice, and then that has the new trait when it regenerates. An example of this is golden rice. And this is present in the tissue, not the actual rice grain. And what that does is it actually introduces beta-carotene into the grain so that you can reduce the vitamin A deficiencies. Okay, so this is very important to help add nutrition into third world countries and so forth. All right, and this is just showing you how that occurred. Another thing is Roundup Ready soybeans and cocaine. Pretty much they have inserted that Roundup Ready gene into the cocaine and the soybean plants. And again, that's so when you spray it on the Roundup on them, it does not die. This is just showing you a little picture of that. All right, we have knockout mice, okay? This is a genetically engineered animal that makes genes inoperable, okay? So it knocks out a gene, all right? And both alleles of the gene are removed or made non-functional. Right? And this is really helpful in the study of cancer, obesity, heart disease, diabetes, arthritis, substance abuse, Parkinson's, even cystic fibrosis. Okay, and this is just showing you how it works. All right, again, the blastocyst is a big ball of cells. Okay, again, you're going to add that in there. You're going to take out that target gene. You're going to knock that out. Okay, and this is just showing you an example in the mice, the actual knockout mouse that occurred. All right, so there are some limitations. They are a little bit lethal, 15% of them, and they typically don't survive as mice as adults. Okay, and some locations on the DNA are very difficult to remove, to knock out, to make non-functionable. Okay, so now we just have a little bit of terminology to talk about. A DNA microarray is either called a DNA chip or genome chip. All right, and this is going to contain microscopic amounts of DNA that is actually in a glass slide or a silicon chip. And this is going to help you tell what genes are turned on in a specific cell. So it's again for more research. Comparative genomics is when you compare the human genome and the human genetics to that of other organisms. Proteomics is the study of structure, function, and interaction of proteins. So a proteome is an entire collection of a species' proteins. And then bioinformatics, is our last slide, is the application of computers, technology, and all of that to study biological information. It's been very helpful in this field of biotechnology. So if you have any questions, please let me know. Thanks.